You're welcome, everyone. Today, I want to talk to you about the Sanger sequencing. Sanger sequencing. Sanger sequencing is also called the chain termination sequence. The reason is because the chain terminates at that point in the sequencing process. Now, when you have thymine, adenine, cytosine, and guanine, as you can see illustrated here, in an amino acid molecule, the third end has a phosphate group in the thymine. The same thing in adenine. Number 3 in the chain, in the 5 carbon chain, has a phosphate group. The same thing when we look at cytosine. The third carbon in the 5 carbon chain has a phosphate group. The same thing in guanine. The third carbon, the third end, has a phosphate group. So the phosphate group is used for linkage of the chain to the next group. The phosphate group this linkage has OH group. So the OH group is the group that permits the linkage. So the O now links up with the phosphate group. Once it links up with the phosphate group, it now continues the chain. As you can see, we have the normal deoxyribonucleotide shown to the left in the upper part here, while we have the dideoxyribonucleotide. The dideoxy has lost the O in the OH at the 3 end. There's a missing OH. So what you have is just the H. Now, this H will not allow for phosphate linkage to the next carbon atom. It will not allow for phosphate linkage in the H. So the chain at that point terminates. If there's a free OH, so that is what Sanger sequencing is all about. When there's a free OH group on the third end, or the third OH, replication will continue when the OH is free. The moment there's no free OH, like we have in the dideoxy, replication sequencing will stop when there's no free OH. What is Sanger sequencing used for? Sequencing is done to identify and to know the sequence, the codons, and to get the codons that are coding for a particular DNA in an organism or in any cell. Now, when you do what is called PCR, before PCR, there are processes. Sometimes you can do colony PCR, sometimes you can do some cleanup before you do the PCR. After you've done the PCR and then you check, you do the gel, you run the gel, then you check the gel for the presence of DNA. Then you clean up the DNA, and then you proceed to set up for sequencing. You send for sequencing. Product of sequencing is the sequencing cannot just continue indefinitely. At a point, it must stop. So that is what the Sanger is for. At a point, the sequence will stop. At a point, as you are sequencing, it will stop. At a point, as you are sequencing, it will stop. Why? Because there's a chain termination. A chain termination has occurred. If there is no chain termination, sequencing would have just continued. So sequencing stops. Then after the sequencing, you arrange all the sequence you have obtained. Then we get the name or the identity of the organism using the database, such as the BLAST, the NCBI tool, and there are other databases that you can use, but the NCBI seems to be the most acceptable. Now, Sanger technique, the DNA to be sequenced. I want to tell you the process of Sanger technique. The DNA to be sequenced is heated such that double-stranded DNA becomes single-stranded DNA. The template strand and complementary strand are formed. primer is annealed to the template strand. This is to allow the addition of nucleotides later. 4 reactions are set up. The template strand with the primer DNA to be sequenced is added to the reactions. DNA polymerase is added. Free dNTP nucleotides are also added to the reaction mixture. One of the dNTPs, e.g. A, either as adenine, guanine, thymine, or cytosine, is radio-labeled 32p atom or 35s atom to determine the DNA sequence later. Modified nucleotides, ddNTPs, are then added to the reaction mixtures. Only one type of ddNTP is added to the reaction mixture. Chain termination occurs after the addition of ddNTP. This is because there is no OH group to attack the next dNTP. The DNA will now be separated based on size using gel electrophoresis. A sample of each reaction mix is taken and put in a separate lane on the gel. Polyacrylamide gel electrophoresis can separate polynucleotide chains differing in size by 1 nucleotide. Electric current passes during the electrophoresis process, and negative charge DNA moves towards positive charge electrode. Smaller fragments will move faster than larger fragments. The gel is dried and X-ray is taken. Labeled dNTP will appear on the X-ray film. The strands will separate and be distinct on the X-ray film based on the type of dideoxy added. So the dideoxy can be ddATP, it can be ddTTP, it can be ddCTP, it can be ddGTP. The chain termination method have greatly simplified DNA sequencing. Chain termination based kits are commercially available that contain the reagents needed for sequencing, pre-aliquoted and ready for use. So this is a simple explanation of the Sanger sequencing. I will talk to you some other time. Bye for now.